Hello, my name's Claire Horwell. I am a professor of geohealth at Durham University in the UK. I'm also director of the International Volcanic Health Hazard Network. In this talk, I'm going to give a short history of health research on the Soufriere Hills volcanic emissions, Montserrat. First of all, why are we concerned about health in a volcanic setting? The Soufriere Hills volcano on Montserrat produces magma of intermediate composition. This means that this magma is quite sticky and sufficiently viscous to erupt explosively sometimes, which can produce inhalable ash particles. Sometimes it erupts effusively, and this means that almost solid rock comes out of the volcano, building a lava dome in the crater. Lava domes crystallize the mineral cristobalite, which is a type of crystalline silica, or SiO2, like quartz. And both of these are known to cause a disease called silicosis and lung cancer in some occupational settings. When lava domes collapse, respirable crystalline silica is generated. The volcano also produces gases which could exacerbate respiratory health problems. Due to knowledge that was gained from the eruption of Mount St. Helens in 1980 in the United States, health research on Montserrat started in earnest as soon as the lava dome started to grow and collapses began. So for those of you not very familiar with the Soufriere Hills volcano, the eruption started on the 18th of July 1995 and lasted in its active phase around 16 years. With the last activity being in 2011. There were 4 phases of eruption, so these took up about 8.5 years of the 16 years that the volcano was active. Now, the first health study was started pretty soon after the eruption began, in February 1996. There had been an increase in gas emissions and a persistent haze around the volcano, and people were starting to notice leaf damage. So some researchers, Allen, Baxter and Otley, came out to Montserrat and measured gas and particulate matter concentrations. Key gases of sulfur dioxide and hydrochloric acid and particulate matter were found actually to be rather low in the ambient air and too low to present an acute health hazard in typical weather conditions. So this was very good news early in the eruption, but air quality got much worse after dome collapses started. So what are the potential health impacts of inhaling volcanic ash and gas? We can divide these into acute symptoms, which happen almost straight away, and chronic symptoms or diseases, which might happen years into the future. So looking at acute symptoms, even healthy people might expect to have some irritation of their airways, so some cough or phlegm or sore throat. But people who already have existing diseases such as asthma, bronchitis, and other pulmonary diseases may well find that they experience an exacerbation of their symptoms. Now, thinking about the chronic diseases that could come, these are really from ash inhalation rather than gas. And our knowledge of these is based on other mineral dust exposures. So not at volcanoes, but more often from mining and quarrying. And we are particularly concerned about some chronic diseases that can be very, very harmful and fatal, such as silicosis, lung cancer, and tuberculosis, all of which are related to crystalline silica, which I mentioned is formed in dome-forming volcanoes. There are other kinds of diseases that are also related to heavy exposure to mineral dust, such as pneumoconioses. But it's important to say that currently there is no scientific evidence of the Soufriere Hills ash generating any of these diseases. But we're concerned about them because we know it's a possibility. So how can we assess the health impacts from volcanic emissions? We can broadly define the different kinds of studies into those that take a long time, perhaps months to decades, and those that happen very quickly, and we get results within hours or up to weeks. So let's look at the more long-term studies first. We have clinical medicine, epidemiology, and in vivo toxicology, which means that you do experiments with animals. The Soufriere Hills eruption, starting in 1995, triggered at least 4 clinical and epidemiological studies, which are summarized here. These were all cross-sectional studies, and 2 of them were on adults. One was on immigrants to the UK, and one was on Montserrat workers, and 2 were on school children. All of them looked for respiratory symptoms through questionnaires, but some also included X-rays and lung function tests. And the results varied, but there was a slight increase in respiratory symptoms for the people who had moved to the UK versus the general UK population, but there could have been a number of reasons for this. There were also a decrease in lung function in gardeners and road workers on Montserrat. And for the children, the children were 4 times as likely to report wheeze in the most heavily exposed areas. And then in the later study with children, the follow-up study, 10% of children had asthma symptoms, but there was also a very high rate of allergy on the island. There was some increased exercise-induced bronchoconstriction as well. But the good news is that the X-rays were normal in all tests that were done. So these were all quite short-term studies. And the question is, have any long-term studies in adults been undertaken to look for the really chronic diseases that might take a decade or 2 to manifest themselves? And the answer is no. These studies are almost impossible to conduct, unfortunately. It is really hard to follow a cohort of people over decades that it might take from exposure to actually falling ill. There's also a very small and mobile or transient population on Montserrat, which makes it particularly hard to follow people, but also to do statistically valid studies. Now, it's a very good thing, but exposure for many people was terminated by their permanent evacuation. So actually, for some people, their exposure was not sufficiently long enough to make these chronic diseases a likelihood. And for these studies, you do have to measure people's exposure to the emissions, and this has been very hard to measure and has not historically been done. So it's hard to work out if people do develop diseases if they are due to exposure to volcanic emissions. And finally, these kinds of long-term studies need a lot of long-term funding, and that can also be very hard to find. So this is why there have not been very many clinical or epidemiological studies done, and no really long-term studies. But we can also use proxies, and one type of proxy is to use toxicology, where we basically use laboratory exposures of animals to volcanic emissions. Now, several studies have been conducted on the Soufriere Hills emissions and in the laboratory. These have been mostly instilling rats or mice and exposing them to volcanic ash. The most important of these studies was by Lee and Richards in 2004. And this showed that the Soufriere Hills ash produced inflammatory reactions in lymph nodes and in the lungs of rats after 49 weeks. So this was a long-term study for a rat. No fibrogenic or silicotic response was observed, which was good news. But it's actually impossible to know how well these findings will translate to humans. So what else can we do to try to assess the potential health impacts? As I mentioned earlier, there are a range of more rapid analyses that we can do. And we have spent a lot of time trying to develop these so that they can be rapidly deployed in eruption crises. I'm going to take you through exposure assessment, mineralogy and geochemistry analyses, and some in vitro toxicology. And I'll come back to syndromic surveillance at the end of the talk. So when we think about these more short-term analyses, we need to think about what we're actually looking for. What are we trying to measure? And that asks the question, well, what is it about volcanic ash that could actually be harmful to the human body? So first of all, let's think about particle size and shape. Particles less than 100 micrometers in diameter can enter the lungs, and I'll talk about that in a second. Fiber-like particles may not be easily cleared. So we do worry about fibrous particles, but as you can see in the image on the right, volcanic ash is usually very, very fine, like talcum powder, at least the type of ash that comes from Soufriere Hills volcano. But fresh ash is very rarely fibrous. So we don't actually need to be particularly worried about long fibrous particles. But let's go back to the size of particles that can actually enter the lungs. This is a really useful diagram. It shows a cartoon of a human hair, and it's about 50 to 70 micrometers in diameter. And we say that about particles sub 100 micrometers in diameter in general can get into the upper respiratory system through our nose and mouth. And again, this is roughly also, you could say, the width of some fine beach sand. Now, further down in the lungs, only much, much smaller particles can get in there. And we call these PM10 and PM2.5. So PM10 is particulate matter less than 10 micrometers in diameter, and PM2.5 is particulate matter less than 2.5 micrometers in diameter. So a PM10 particle is roughly a fifth the width of a human hair, and a PM2.5 particle is about a 20th the width of a human hair. So these are incredibly small particles which can get actually into the human lungs. So actually size really matters. Coarse particles of ash cannot enter our lungs. We really are looking at very, very fine particulate matter. And particles around 100 micrometers get lodged in our throat and in our mouth and nose. And then particles of around 10 micrometers will deposit in our bronchi. And you can see here the little pink line that I've drawn here to show where these particles might deposit themselves. And this is the area where we think that acute asthma flare-ups will happen from exposure to ash as it irritates the lining of these tubes in our lungs. But we then have a system of tiny, tiny branches in our lungs called bronchioli, and then alveoli right at the ends, little sacs where gas exchange happens. And as you can see in this electron micrograph of the alveoli up here, these are tiny, tiny, tiny sacs, only about 5 micrometers in diameter. And in order for our gas exchange to work properly, we do not want these to be full of particles. So if we have particles of sub 4 microns or 2.5 microns, which can get deep into these alveoli, then that can become really problematic. And this is where potentially these chronic diseases like silicosis might develop if we cannot effectively clear these from our lungs. So how do we find out if our volcanic ash is small enough to get into our lungs and is actually inhalable? There are two main methods that we use. One is through air quality monitoring, which can also be called exposure assessment. And this looks at how much fine particulate there is in the air, either through ambient or environmental monitoring or through personal monitoring. And in the image on the right, you can see Alison Searle, who was at the Institute of Occupational Medicine in Edinburgh, conducting personal monitoring next to a gardener on Montserrat. But often personal or environmental monitoring is not happening. And if that's the case, what we tend to do is to actually collect settled ash off the ground and then analyze it to see how fine the particulate in the ash is where it fell. So if we just look at the exposure studies that have been done at Soufriere Hills volcano, Alison Searle and her team conducted a number of studies and published this landmark paper here, looking at environmental concentrations of particulate matter and finding that, not that surprisingly, that concentrations were highest in the south, nearest the volcano. She also did personal exposure concentrations of people in different occupations. And in particular, she found that gardeners were very highly exposed. to volcanic ash. And she also looked at the concentrations, personal concentrations of children conducting different activities at school. Now, if you're particularly interested in exposure assessment studies, I've also put links to a couple of other articles that have been published on Soufriere Hills air quality monitoring and exposure assessment. So as mentioned, if air quality monitoring is not taking place, then you can also conduct particle size analysis of settled ash deposits. So you collect a sample of ash, and then you can either sieve it, or you can use a more sophisticated technique like laser diffractometry to assess the quantity of inhalable and respirable material in your ash. And I published a paper in 2007 where I compared the Soufriere Hills ash, which you can see in this red box, with other samples from around the world. And if you just have a look at the 10 micron column here, so this is the amount of ash less than 10 micrometers in the samples analyzed. And you can see that for dome collapse samples, we have around 23% of the ash is sub 10 microns in diameter. And this is quite a lot of the ash, and it compares favorably with other dome collapse ash from around the world, for example, this sample from Merapi, but also with eruptions that are extremely explosive, such as the Mount St. Helens eruption in 1980. Now, lesser explosive eruptions, like the kind of Vulcanian explosions that you have in Montserrat, tends to generate less particulate matter, in sub 10 micron and respirable fractions. As you can see here, only 13% compared to 23%. So we regularly use this kind of analysis to quickly tell us roughly how much respirable material we have and whether it is a potential respiratory health hazard. So we talked about particle size and shape, and now we're going to move on to this mineral crystalline silica, which I've inferred in this talk we are quite concerned about because we know from other settings such as mining and quarrying that exposure to crystalline silica can cause these diseases such as silicosis and lung cancer. So we know that the Soufriere Hills eruption is generating or has generated the mineral cristobalite. So what you can see here are some images of cristobalite particles. Here is this beautiful crystal. It's only about 10 micrometers in diameter, so more than small enough to actually get into the human lungs. But here we see it actually in a piece of dome rock. This is not a piece of ash. And if we were to take a slice through this dome rock, we actually see that we're looking at this crystal actually sitting in a kind of bubble in the dome rock. So what actually happens as you have the Soufriere Hills dome sitting here in the crater of the volcano is that slowly over time, the dome is cooling and it's also degassing. So we know that we can see gases coming out of the dome all the time at the volcano. And as these gases percolate through the dome, they're moving through little cracks and pore networks and bubbles, and they basically then come out of the dome. And that's what we see at the surface. But in the process of moving through the dome, slowly over time, little crystals of crystalline silica are basically formed as kind of silica vapors are deposited on the walls of these cracks. There's also a second way of producing crystalline silica in the dome, and this is through the volcanic glass, which makes up the lava, actually slowly crystallizing, and that's called devitrification. And as that happens, it crystallizes into crystalline silica and also a mineral called feldspar. So we have a lava dome, which is sitting there growing cristobalite, type of crystalline silica. And we know that these lava domes are highly unstable, and sometimes they collapse, generating pyroclastic flows or pyroclastic density currents, as they are also known, which travel down the valley, such as the Tar River Valley here. And as they do that, they kind of loft the fine-grained particles that are being generated as the dome is being pulverized. And this lofting plume of fine-grained particles, which is now volcanic ash, then deposits on local areas around the volcano. So this is how people become exposed to the crystalline silica. Now, I've mentioned several times that we are worried about a disease called silicosis, which comes from crystalline silica exposure. Silicosis is a fatal, inflammatory, progressive scarring lung disease, which takes 10 to 20 years for it to manifest itself. So I showed you this image here of the electron micrograph of alveoli in our lungs with particles inside. And here we have a kind of cross-section through a healthy lung. And you can see on the left-hand side here that the lung has got lovely big alveolar sacs, and they're clear, and the walls between them are very thin, and so gas exchange can happen easily. But if you start to get a lot of particles in this part of the lung, then as you can see in the middle photo, the walls start to thicken and the alveoli become smaller. And finally, as this continues over time and the body tries to get rid of these particles, basically through reactions, you can grow these kind of collagen nodules or silicotic nodules, which basically start to fill up the lung, take away the alveolar spaces, and overall make it very difficult to breathe and to function. So unfortunately, silica has also been classed as a lung carcinogen in 1997. But when the World Health Organization, through their IARC group, classified crystalline silica as a lung carcinogen, they made a very important caveat. They said that carcinogenicity to humans was not detected in all industrial circumstances studied. Carcinogenicity may be dependent on inherent characteristics of the crystalline silica or on external factors affecting its biological activity or distribution of its polymorphs. What this basically means is that if you are exposed to crystalline silica in large amounts or over a long time, it does not necessarily mean that you will develop lung cancer. And there's something going on with the crystalline silica which is influencing this. This became known as the crystalline silica enigma. And so the question is, and this also applies to silicosis, is the Soufriere Hills crystalline silica and crystalline silica at other similar volcanoes capable of causing these terrible diseases? So going back to the cristobalite at Soufriere Hills, it was first discovered that the dome collapse ash contained cristobalite in 1996. So the British Geological Survey did a test and found that 25% of the sub 10 micron ash was crystalline silica. Now, this was a really high amount and far higher than previously measured at Mount St. Helens in 1980. So this triggered another landmark study conducted by Baxter and colleagues, where they measured the crystalline silica content of different kinds of ash from Soufriere Hills and using a few different methods. And they corroborated this result, and they saw between sort of 10 and 27% crystalline silica in dome collapse ash compared to volcanic explosion ash, which had much less crystalline silica in it. So much more research was then conducted on this crystalline silica. I started my PhD a year later and worked on this extensively. And what we were able to find out was that crystalline silica-rich ash occurs when you have dome collapses and in periods of normal extrusion rate of the lava, then you seem to get quite high amounts of crystalline silica, more than about 7 to 8%. Whereas in times when the lava dome is growing extremely rapidly, as you can see in the upper diagram, we do see an increase in crystalline silica over time, depending on how long a piece of the dome has been there. But actually, it's much less when it's growing rapidly than when the lava dome is growing quite slowly. So we also see high crystalline silica at times when the Soufriere Hills volcano is ash venting through a lava dome, and also when there are explosions through the lava dome. But in times when ash venting happens without dome incorporation, and the same with the explosions, then we get less cristobalite. So I mentioned that one way that we can also quite rapidly analyze volcanic ash is by doing another kind of toxicity study called in vitro toxicity. And here, instead of looking at animals in the laboratory, we expose cells to volcanic ash. And so this can be done much more rapidly and also much more ethically. And there are a few different kinds of cellular responses that we can look for when we expose cells to volcanic ash. The first one is a cytotoxic response, which basically means that the cell is either dying or letting out signals to show that it's very in a lot of trouble. And in the top graph, you can see an analysis of cytotoxicity with a measure of cytotoxic response on the y-axis. And on the x-axis, you can see increasing dose that was given to the cells. And the black line at the top is DQ12, which is a quartz standard of very high toxicity. And we can see here that the cells did react and they were in trouble, and that increased with dose. But down here, we see that the ash sits way down. And in fact, the other black line with the squares shows a basically negative control of titanium dioxide. And the ash was less than that. And Soufriere Hills, SHV, is in there amongst them. So the cells that we were looking at did not really respond to having volcanic ash put on them. They were not in trouble. However, sometimes we do see that cells let off what's known as an inflammatory response, which can be a precursor to cancers. And so again, here in the bottom diagram, we have a number of ash samples, and we also have the DQ12. This time, the increasing dose is actually shown as bars of different colors. And so you can see that the DQ12 is causing the cells to have a high inflammatory response, especially at high doses. But we do also see a significant inflammatory response for Soufriere Hills ash here, and also ash from Montserrat volcano. So there is something going on in these toxicity studies, but it's not as straightforward as we might expect from other kinds of quartz. So why might the Soufriere Hills ash or cristobalite not be as toxic as we might expect for a crystalline silica? Well, we talked about this crystalline silica enigma. There might be something going on actually inherent within the crystals themselves. And that's exactly what we found. So when we actually looked at the composition of the individual crystalline silica particles or crystals, we actually found that when we looked at quartz, as expected, it was almost 100% SiO2. But when we looked at cristobalite, and you don't need to worry about the different kinds of cristobalite here, you can see that there's up to 3 weight % aluminium in the structure, the chemical structure of these cristobalite crystals, which should be just SiO2. Now, actually, it was previously known that cristobalite has got quite an open crystalline structure and can actually take in aluminium and sodium ions into the structure. And that seems to be exactly what has happened in Soufriere Hills cristobalite. And also, we've now discovered in any other kind of cristobalite that we see in different volcanoes around the world. So there seems to be something that, is impure in these cristobalite crystals, and maybe this impurity could impact toxicity. So we actually tried to find out if that was the case in the lab. We actually grew cristobalite in the lab, and as it was growing, we either kept it pure or we added some aluminium and sometimes sodium as well to see if this impacted toxicity. So this is called doping. And then we checked the cytotoxicity for these different samples with cells. So here again on the right-hand side, we have our DQ12 sample, and this is the toxic quartz, and it was almost 100% cytotoxic. And here in white, you can see 3 bars. These are for the pure cristobalite samples that we generated, and they are quite cytotoxic, more than 60% roughly. But then if you look at these grey bars, as we added aluminium into the mix, so at 1%, 2% and 3% respectively, you can see that the cytotoxicity quite rapidly drops. And this shows us that actually it seems like the aluminium in the crystalline silica actually does sort of dull its toxicity. So this was a very important result. But we also found another reason why the dome cristobalite may not be as toxic as expected. Now, I want you to take yourself back to the slide where I showed you what dome collapse looks like and what the cristobalite crystals look like. And they're sitting, here they are, inside this kind of vug or kind of hole inside the lava dome, surrounded by lava dome material and actually attached to it as well. And so you can imagine that when this lava dome collapses, you wouldn't necessarily expect to get pure crystals of cristobalite. And what we actually see is that a pure crystal like the one on the top left here is really very rare. And usually what we see is just what we would expect, a crystal still stuck to the dome, as you can see in image B here. So this is kind of the dome material on the left and the crystal on the right. In the bottom left, you see in image C, a bit of crystalline silica in the center of this particular particle, and you see dome material all around it. So this was probably a bit of devitrified crystalline silica. And then on the right-hand side in image D, you see another nice crystalline silica crystal almost completely surrounded by bits of ground mass from the dome. So when we think back to what IARC said, they said there might be inherent characteristics of the ash, of the crystalline silica, which we've shown. There are impurities in the silica itself, but also external factors which might moderate the toxicity. And so here we see that we've got this bits of dome still stuck and surrounding the cristobalite, and this is going to impact the reactivity of the surface of the particle with the lung, and we think reduce it. So does this mean that Soufriere Hills ash isn't going to cause silicosis or lung cancer? Well, we have some reasons why it might not, but we just do not know because there is insufficient clinical evidence. So this means that we still need to take a precautionary approach and assume that it could happen. And in fact, when I first presented this talk on Montserrat, somebody in the audience told us afterwards that they thought they knew of several people who had, in fact, died of silicosis. So this needs to be followed up. So what other tools have we got to try to understand the health impacts of volcanic ash? Maybe we could use other types of particulate matter as a proxy for volcanic ash. So in 2012, the World Health Organization produced an important report called Review of Evidence of Health Aspects of Air Pollution, or REVIHAP. And this report was following a meta-analysis of many studies, and it showed that both short and long-term exposures to fine particles like PM2.5 can cause respiratory and cardiovascular morbidity and mortality. So that means diseases like cancer and death. So if we were to apply what they found, which they said was applicable not only to urban pollutants, but to other kinds of particulate matter like crustal materials, then we have to assume that volcanic ash could have the propensity to do this as well. And in fact, they also linked particulate matter exposure to many other adverse health outcomes in the human body. And more evidence is emerging of this every day. So we looked at whether we could carry out health impact assessments of volcanic ash using outdoor air pollution methods and risk estimates. And we concluded that this could be done with caution, but we're still really lacking the evidence. And we also know that volcanic ash has a very different composition to, say, diesel exhaust. So can we do our own risk assessments of volcanic exposures? So in fact, one of the very first risk assessments of its kind was conducted at Soufriere Hills, or Montserrat, on Soufriere Hills ash. And this team used data from Alison Searle's exposure work, the kind of physicochemical and toxical ash analyses that we were doing, and also the clinical and epidemiological studies. So basically all of the hazard assessments were employed that had been done, and these were then fed into a quantitative silicosis risk assessment. And this was a modelling exercise that was completed in 2003. And the key findings were that if an average adult on Montserrat, was exposed continuously to volcanic ash for 20 years, so that would take us up to 2023, and actually most of the exposures were finished by around 2011. Then someone living in the north of the island would expect a 0.5% risk of developing silicosis, and someone in Cork Hill would expect a 2% risk. Now, the risk was higher for occupational groups, and so for gardeners, it was up to 4% risk of silicosis, but the risk was even higher for children. And in fact, it was up to almost 9% for children who would have been living in Cork Hill. But of course, Cork Hill has been evacuated for many years. So the risk for most children on Montserrat is considerably lower, as you can see from the rest of the table in the bottom there. So let's move away from crystalline silica and think about what else could potentially be harmful in volcanic ash. So one aspect which is not often thought about is the reactivity of iron in volcanic ash. So iron is known to undergo a reaction with hydrogen peroxide called the Fenton reaction, which generates something called the hydroxyl radical. Now, hydroxyl radicals in the lung are capable of damaging DNA, and this potentially could lead to the development of inflammation and lung cancer. So does this happen for Soufriere Hills ash? So actually my PhD work was the first to recognize that the iron in ash could potentially have this biological pathway in the lungs. And so we tested the surface reactivity of Soufriere Hills ash to see if it could generate this hydroxyl radical. And in the top right graph, you can see that we compared the toxic quartz standard. In this case, it was one called Minusil quartz rather than DQ12, but very similar, compared to various ash samples. And this is hydroxyl radical generation. And you can see that indeed, the Soufriere Hills ash, which of course is not pure crystalline silica like the toxic quartz standard, it has other minerals in it as well, could produce substantial amounts of hydroxyl radicals. Or so we thought at the time. In the graph underneath, you can see that we then analysed lots and lots of different ash samples, and actually Soufriere Hills ash, being an intermediate magmatic composition, does not have a huge amount of iron in it compared to some other types of volcanic ash like basaltic ash. And so actually when we looked at total iron release for lots of different ash samples against hydroxyl radical generation, we saw that Soufriere Hills ash was way down here, close to zero, between 0 and 100, compared to, for example, ash from Etna, which is a basaltic volcano, very iron rich ash, which generated far more hydroxyl radicals and could release far more iron into the lungs. So we also are concerned about leachable elements, and there are a range of elements which could leach into lung fluids or water supplies from the surface of ash particles. And one of the elements we're most concerned about is fluoride. You can see here an image of a child with teeth that have been affected by fluoride, leading to the disease fluorosis. This image is not from Montserrat, to be clear. And in fact, we do not think that there is the soluble elements on Montserrat from the ash are very concerning. But having said that, very little work has actually been done. And this is something that should be done in the future. So we don't really know how soluble elements from Soufriere Hills ash might affect respiratory health or water quality. However, what we do know is that usually in an eruption, when ash enters the water supply, it usually makes it cloudy and taste bad, rather than actually causing water quality guidelines to be exceeded. There was some work done on leaching of volcanic ash by Marie Edmonds, not for health hazard analysis, but she did show that the ratio of different elements, including sulfate, chloride, and fluoride, does change depending on whether the ash came from dome material or from other explosive eruptions. So what about next time when Soufriere Hills erupts? And we must remember that the current eruption is not actually finished. It's just that there are no external signs of activity for the past decade. So a key thing to think about will be preparedness. It's time now to plan, to know which agencies and collaborators would be responsible for which analyses in the future. Now, I mentioned early on that we can do a rapid analysis called syndromic surveillance, and I haven't explained what this is. It's effectively rapid epidemiology. It's usually carried out to check for communicable diseases in crises, but it can also be adapted to check for health impacts related to emissions, volcanic emissions in the short term. So setting up syndromic surveillance and having that system available in advance is really important. Working out whether and how you could do water quality monitoring and air quality monitoring, and also ash analysis. So that's the physicochemical and toxicological analyses that I have taken you through. It's a really good idea to get baseline data in periods of quiescence. So right now being a very good moment. And in fact, this is why we have recently set up an air quality monitoring project on the island in May 2023. It's also a good moment to consider what equipment would need to be on island to support all this monitoring. Of course, disaster management folk will be considering evacuation and shelter plans, but it's also a good idea to think now about stockpiling respiratory protection and clean water if that might be needed. We did a large study which showed that, just as for COVID, N95 grade masks were absolutely the best kind to protect from volcanic ash, and that surgical masks, while they filter relatively well, do not fit to the face and do not really protect people very well from ash exposures. And I can provide you with much more information on that if you would like. Another key aspect of humanitarian response is to reduce humanitarian waste as much as possible. And this means trying to avoid bringing bottled water to the island, especially as there is no there's no recycling on Montserrat. And this was quite a big problem during the St. Vincent eruption in 2021. You can see the image on the right, where a lot of waste was brought onto the island unnecessarily and then had to be cleared. So you're probably wondering how to go about all of this. Well, the International Volcanic Health Hazard Network can support you. This is what we are here to do. We founded IVHHN in 2003. We support preparedness and crisis response and also public communication. We have a website at www.ivhhn.org, and you can see that on the left-hand side in the image. We have a page, you can see right at the bottom, on crisis management, and that page is shown in the right-hand image, where we have briefing notes for eruption management, which can help you prepare for an eruption and cope with the eruption while it's happening in terms of the health response. We have also designed protocols for rapid analysis of volcanic ash. So these are the physicochemical and toxicological analyses. These are off-the-shelf protocols that have been tried and tested and published. As you can see here, we've got examples of protocol design and testing, and then enactment of those protocols in different eruptions. And this allows standardization of methods and a confidence in methods as well, and also allows us to then compare among volcanic locations so that we can compare, how bad one situation is compared to previous knowledge. We have also designed standardized epidemiological protocols, and these are especially useful where syndromic surveillance is not already in place. So many epidemiological studies have been conducted following eruption crisis, but unfortunately they often miss the very critical periods of highest exposures soon after the eruption onset, while study methods are being developed and approved. So this is why we developed these off-the-shelf protocols so that hopefully they could be enacted much faster. We have two protocols. The basic protocol assesses the respiratory and other health impacts from acute exposures, and the cross-sectional protocol assesses the extent to which volcanic exposures cause the adverse respiratory and other health effects. Advising relatively simple and inexpensive methods, the protocols are applicable in all volcanic contexts, regardless of resource availability or health record systems. And we really hope that consistent use will generate a strong and comparable evidence base of the health impacts of eruptions. We are in the process of trying to embed these protocols within existing crisis response plans in Latin America and Caribbean countries. So IVHHN also has a suite of public communication products. These are in the form of pamphlets with a lot of information, booklets and wrap brochures and leaflets with kind of more critical information, and also posters that can be put in health centres and local public spaces with just the key information in them. And these are on the health hazards of volcanic and geothermal gases, how to prepare for ashfall, health hazards of ash as well, and also how to protect yourself from breathing ash. And these are accompanied by a suite of videos as well. So these can all be found online on the IVHHN website, and they can be adapted for use for your particular agency by adding your logos, for example. So the take-home messages from this talk is that no studies have identified any chronic disease on Montserrat yet. Evaluating disaster health risk in volcanic environments is extremely challenging. As I've shown, impacts could emerge or continue for years or even decades after an eruption starts. So a precautionary and planned approach to reducing exposure and risk is necessary in order to mitigate health impacts. I'd be very happy to discuss this with you more, anything that I've said here, and I hope this has been a useful record of the health research done on Montserrat. Thank you very much.